This video is going to help explain the bacteria and archaean chapter. It is going to mostly be about bacteria. In fact, I don't know that I'm really going to talk about archaeans all that much during this chapter. But this is going to be about our earliest forms of life that have been on the planet for the longest period of time. They consist of prokaryotic cells, and these are the two domains that do that. So first thing for us to do is discuss biofilms and why they're medically important. Biofilms are something that, whether you know it or not, you have been exposed to them and you have them growing in you. So every time you go to the dentist and they have to take those wonderful metal hooks and just scrape at your teeth, they're scraping a biofilm called tartar off your teeth, and that tartar consists of a bunch of different species of bacteria plus this layer of usually a carbohydrate that they're kind of gluing themselves to your teeth so that it's harder to remove them. That tartar is more difficult to remove because they've created the community. That's why they have to use the metal hook to remove it. And if it doesn't get removed, that can lead to periodontal disease, dental cavities, tooth loss, and bad things happening generally. The bacteria do it essentially so they can create this ideal living condition where they're making sure that they get a constant supply of nutrients and their wastes can be removed and conditions just stay right in there. It just usually doesn't work to our benefit when they create a biofilm because it's harder to kill once they've created a biofilm. Next up. What are prokaryotes? These are anything that is made with a prokaryotic cell. We've discussed the two different cell types a couple of times before, but just remember prokaryotes are considerably smaller in real life than eukaryotic cells are. They don't have any of the membrane-bound organelles, no nucleus, no rough ER, no smooth ER. They don't have mitochondria or Golgi apparatus. They don't get chloroplasts. Instead, everything's just kind of thrown into the cell, and there's no compartmentalization within that cell, so everything is just flung in there. The two domains of life that consist of prokaryotic cells are the domain bacteria, which is mostly what we're going to be talking about today, and then the domain archaea, which usually consists of organisms that we like to consider extremophiles. They live in places that are really hot or really salty or really acidic. We're not going to spend a lot of time talking about them because they're not really all that relevant when it comes to medical health, really. Next up, you have this picture to label. Remember, you can pause this so that you can get all the labels that are on there. This is just a picture to show you what a cell that is prokaryotic can have. They don't have to have all of these components, but it's possible. So first off, they do get cell membrane or plasma membrane or cytoplasmic membrane. It has multiple names, but their cell membrane is nearly identical to yours. It's still a phospholipid bilayer with proteins scattered throughout it. Outside of that, they have a cell wall, which is in purple in this picture. Their cell wall is made out of a unique molecule called Peptidoglycan. Plants have a cell wall, but they don't use the same molecule to make it. Same for fungi. They use a cell wall, but they don't use the same molecule to make it. Outside of the cell wall, they've labeled this the glycocalyx. Some cells have a slime layer, some cells have a capsule. There's different components that can make that up, but usually whatever's outside of the cell wall is there to help protect the bacterial cell from the environment that it's living in. Next, everything inside the cell is all cytoplasm. Inside the cytoplasm, we've got one single circular chromosome in most of our prokaryotic cells. It's hard to tell that it's a circle because it's usually packaged up a little bit, but it is one single chromosome. A lot of times some bacterial cells will have additional pieces of DNA that are small and circular. Those are called plasmids. We'll talk about those again here in just a minute. These little hair-like extensions that are coming out, those are called pili for plural. A pilus is singular. Those usually help the cells stick to whatever they're next to so that they don't get washed away or pulled away by whatever happens to be moving over them. Some prokaryotic cells will get flagella, which is plural. Flagellum is singular. This one has 4 of them. If they have flagella, that means they're going to be able to move around in whatever their growing medium is. Usually, though, it means they're going to be swimming, so we're going to need an aqueous environment in order for them to do that. So those are the main components that a typical prokaryotic cell can have. Next up, describing the function of the following. So the nucleoid is the region in that cell where their one chromosome is going to be found. Keep in mind, it does not have an envelope around it. It is not the same thing as a nucleus. It's just that spot where that chromosome can be found. So it's not protecting the DNA at all. Plasmids are those extra pieces of DNA that the cells can have. Since bacteria can't do sexual reproduction, it's still important that they're able to change their genes if the environment changes. And so one of the things that bacteria have been able to do to help with that is exchange plasmids between them. So let's say that this cell has this wonderful gene on here that allows this cell to utilize some nutrient that this cell can't make. If it swaps that plasmid with this other cell, well, now that cell can also use that nutrient. And so it's a way for them to sort of share behaviors or share enzymes or genes or things that help them be more suited to a particular environment. Ribosomes, they do the same thing in them that they do in us, so they're still for protein synthesis. The little side note for them, though, is that their ribosomes are different anatomically than ours. They are smaller than ours. Anything that's different between our cells and prokaryotic cells, that is a target that we will use for antibiotics. And so a lot of our antibiotics actually work by disabling the ribosomes that are present in prokaryotic cells because theirs are smaller. It attacks theirs specifically and leaves ours alone so that we're still okay to do whatever it is we're doing. The cell wall. So this picture I pulled from a micro textbook to show you what's up. So this is the phospholipid bilayer, the cell membrane. Outside of that, there's this layer of blue and purple balls. That's peptidoglycan, and it forms this additional shield outside. So this is the cell wall. This picture is also showing you a unique feature that a certain group of bacteria called gram-negative bacteria have, where they have an additional membrane outside of that cell wall. That's unique to them, though. Not all organisms do that, but it's an important feature that they have from a medical standpoint. But this peptidoglycan is what makes the cell wall for them. Once again, since that's a unique compound that only bacteria use, that's another target that some of our antibiotics are going to use so that we can hurt them without hurting you at the same time, barring an allergic reaction to the drugs. Next, the cell wall helps to maintain the shape of an organism, and there's three basic shapes that you can find. There's some additional ones too, but these are the three that we're going to ask you to remember for our purposes. So anytime you see coccus, it means that the cell is ball-shaped or it's spherical or it looks round under the microscope. So all these yellow cells up here at the top, they're all coccus-shaped. Bacillus means that the cell is going to look kind of rectangular when you see it on a 2D field, but they're column-shaped when you see them 3D. So these cells that are pink down here at the bottom, they are bacillus-shaped. The third option, anything that kind of looks spiral-shaped or corkscrew-shaped, we're going to call that spirillum. Again, there are some different options, but you guys are non-majors, and so we just want you to know ball-shaped, rectangular, and then corkscrew-shaped. So coccus, bacillus, and then spirillum are the ones that you guys are going to have to know. Next, there are some prefixes that we can use to describe what an organism looks like under the microscope. If you see the prefix staphylo, it means that the organisms are clustering together under the microscope slide. So what you do is you take the prefix and then you put it together in a lot of cases with the shape of the organism. So if you see something that is ball-shaped and clustered, it means that there's Staphylococcus. We're going to talk about Staphylococcus aureus at the very end of this chapter, but this is Staphylococcus aureus, the organism that causes staph infections. Notice it's ball-shaped cells that are clustered together, so that's why it's called Staphylococcus. This particular organism tends to cause wound infections, and that's what this person has on this side. Next, the prefix strepto means that the cells are going to be chained together, sort of like a pearl necklace or a rectangular necklace if we're talking about a bacillus, because they can do both, well, this one as well. So this is a chain of ball-shaped cells, so that's Streptococcus. Streptococcus pyogenes is the organism that causes strep throat, which is why I'm showing you this picture. So if we were to take a swab of this and then view it under the microscope, this is what we would see, that little chain of ball-shaped cells under the microscope. So Streptococcus just means chain of ball-shaped cells. One of the things that we can do to help us look at organisms under the microscope is stain them, and because microorganisms tend to look very similar, we have created things called differential stains that stain organisms different colors. One of the first ones of those ever described was the Gram stain. Hans Christian Gram is the guy who stained things as he was looking for the causative agent for some pneumonia that was hitting the hospital that he was working at. It uses 4 different steps, and it uses 2 different stains, a decoloring agent, and then a mordant. You don't have to stain things for yourselves or anything, but this just kind of shows you the steps that have to happen. But what it does at the end is it makes organisms either look pink or purple. If something is purple, it's gram-positive, and so we know that this Staphylococcus is a gram-positive organism, so it could be that Staphylococcus aureus we were talking about a second ago. These bacilli, or rod-shaped cells out here, they're gram-negative because they're pink. And as you notice, most of them tend to occur singly, and so they don't really do staphylo or strepto arrangements in this particular case. This could be something like E. coli under the microscope because it's a gram-negative rod. So by gram staining, we can tell the difference between cells that are on there and then help identify a particular infection that a person has. One of the things that's starting to happen is antibiotic resistance, where bacteria are resistant to antibiotics, and so it's very tough for us to treat certain infections. What we can do now, and what doctors should do, is grab a culture of the organism that the person's infected with, and then try to figure out what that organism is and which antibiotic would be the best to prescribe to that person to kill that organism so that their infection goes away. And a gram stain is one of the things that they can do to help identify that organism. Next up, what are pili? Is plural, pilus is singular. So these are those little Velcro-like extensions that I was describing as we were labeling the picture. They can help the organism stick to whatever surface they're growing on. And in some cases, some of them are going to use a particular one to exchange plasmids between cells. And so it can also sort of kind of be like the bacterial equivalent of a penis that's dropping off some genetic information between cells. What is a flagellum? It is that little whip-like tail that they can use to help them swim around on a surface. Not all bacterial cells have a flagellum. Some have just one, some get one on either end, some get a bunch of them. All the cells have different numbers and different amounts in different locations, but if they have at least one, they can swim around on the medium that we're growing them in. If you're unfamiliar with that word aureus, it just means watery, so it means they can swim. They can't really use this to crawl around super well. It's got to be a watery environment. What is an endospore? It is a very difficult to kill cell that bacteria can use. It's kind of analogous to a seed in a plant. It's a dormant cell that's not metabolically active, it's not reproducing, but it's not dead either. And if the conditions get right, the endospore can germinate, sort of like a seed can germinate to make a plant. When the endospore germinates, it's going to create a vegetative cell that can now potentially cause infection if it happens to be a pathogenic organism. The example that I'm giving you off down here, botulism is something that everybody used to know about, and then when people stopped canning their own food or preparing their own stuff, that knowledge sort of went away. And what's kind of frightening about that is there are people, I'll call them hipsters just for fun, that are getting back into the swing of doing things old school like that, and so they're canning, and if you aren't very careful when you can something, if a specific species of bacteria called Clostridium botulinum gets into the food as you're canning it and you don't heat it appropriately, it's going to form endospores that now can't be killed. So even if you take that food out and you cook it afterwards, that doesn't kill the Clostridium botulinum endospores nor the toxin they produced as they were in the food. And botulism paralyzes the muscles, which means you will stop breathing and you will die. It's not just like, oh, I got diarrhea and it sucks for a day. You die from botulism. And so if you ever do any home canning on your own, please make sure you follow all precautions to make sure that you're sterilizing to the best of your ability to kill anything that might have been present on the food that you were sampling or cooking. The next few terms that we have describe sort of the nutrients and things that the bacteria are going to need in order to survive. These two terms are all about how an organism gets the organic molecules that they need to grow their cells. They aren't just exclusive to bacteria. In fact, we talked about these in relation to plants and animals too. But there are autotrophic bacteria known as cyanobacteria that are photosynthetic, and so they can make their own organic molecules. There's also a bunch of different species of bacteria that are heterotrophic and have to get their organic nutrients from whatever medium they're growing on, even if that medium is you. Next two terms describe how the organism is going to get its energy. So anything that is phototrophic is going to get its energy from light. So those cyanobacteria I mentioned earlier that were autotrophic, they're also phototrophs because they use light so that they can make those organic molecules. Sometimes those two words actually get thrown together so that if you ever see the term photoautotroph, it means they're getting their energy from light and they're getting their molecules in their inorganic state, and then they're using that to make their own food so that they can make their own energy. Chemotrophs, on the other hand, have to have molecules to get that. It's usually going to be organic molecules, but there are some really weird bacterial species that can use stuff like hydrogen sulfide as an energy source. So even though we usually use these terms again in reference to plants and animals, these terms both apply to bacterial cells as well. Next up is about oxygen requirements. If you see the term obligate aerobe, it means that you are obligated to use oxygen. Like you are obligated to use oxygen. If you get cut off from oxygen, you become cyanotic, like this dude, which means you're blue. and then you die. And so a lot of bacterial species work this way too. So if we try to grow them in the lab, it means we can't grow them in an airtight container because then they would suffocate just like you would suffocate. Obligate anaerobes are the exact opposite. They cannot be near oxygen because oxygen actually kills them. When we grow these in a lab, we have to have some very stringent requirements. We can use something called a candle jar that will suck all the oxygen out of the environment as they grow. Labs that deal with these in higher quantities actually have these anaerobe, sort of like a fume hood, where they have to stick their hands in through those gloves and then work with the organisms inside there. Because if this gets breached and oxygen gets in there, it kills all the organisms that you were trying to grow. There's some organisms that live in you, specifically in your gut and towards the back of your mouth, believe it or not, that are obligate anaerobes, and oxygen would kill those organisms. Facultative anaerobes kind of get the best of both worlds. They prefer to have oxygen available because they can make more energy through cellular respiration, but if you deny them oxygen, they don't die, they can continue to grow because they'll switch over to a fermentation pathway. We dealt with a lab last semester with yeast. We did the fermentation lab where we looked at the carbon dioxide that got produced. Yeast is a facultative anaerobe. It prefers to have oxygen available, and if it does, you don't get alcohol and you don't get your bread rise quite the same way. But if you deprive it of that oxygen, you will actually get them to switch to an alcoholic fermentation pathway, and then you can make your wine and your beer and your liquors and things like that. And so they're capable of going either or, with or without oxygen, depending on what's going on in the environment. How do antibiotics kill bacterial cells but not ours? So this picture kind of shows you, once again, we want to target things that are different between our cells and their cells. So thing number one that we can do, they have a cell wall and you don't. That means you can attack their cell wall without hurting you at the same time. So beta-lactam drugs are things like penicillin or Keflex or cephalosporins or methicillin, pretty much anything that ends in illin, it's a beta-lactam drug. Now, there's a lot of people out there who are allergic to beta-lactam drugs. That's not because it's attacking your cell wall, because you still don't have cell walls, but it's a drug that has upset your immune system, and so you've started to make these antibodies called IgE antibodies against the drug. That's a different process that I don't really want to get into a lot. Just know that all of these drugs that are on here, vancomycin and bacitracin as well, they attack the cell wall. When you attack the cell wall, it means that cell can't maintain its shape and it doesn't have that extra layer of protection. So usually what happens is water flows into the cell and then it pops the cell open. So that's how those drugs kill bacteria. Next, I'll do nucleic acid synthesis. Fluoroquinolones and rifamycins like rifampin. These drugs specifically work on enzymes that bacteria use to either replicate their DNA, because they use slightly different DNA polymerases than we use, or make their RNA during transcription, because again, they have different RNA polymerase than we do. And so we can target their different enzymes so that they can't copy any more of their DNA. This often does not kill the cell, but it prevents them from replicating so that your immune system can then go wipe them out faster because they're not reproducing anymore. Cell membrane integrity. So their cell membrane is very similar to yours. But that said, they do use a slightly different thing to help maintain fluidity of their membrane. Polymyxin B is something that is in a lot of topical creams, and that's because since their cell membrane is so similar to ours, this drug is actually really toxic to our cells too. So we don't ever ingest this or put it in through IV. We can only rub it on the outside because it's fairly toxic to you as well. Next up, metabolic pathways. So folate is a B vitamin. These two drugs block two different steps in the pathway for a bacterial cell to create folate. If they can't make folate, they can't reproduce themselves. This drug doesn't hurt you because you couldn't make folate anyway. You have to get that in the foods that you eat, and so that's a biosynthetic pathway that you were unable to do anyway. Last option, protein synthesis. Most of these are going to be blocking their ribosome that I mentioned earlier had a different size than your ribosomes, and so we can selectively hurt theirs. Some of these do have some pretty nasty side effects for human beings, including the chloramphenicol right here, which can cause pernicious anemia. The reason these tend to have a higher side effects is while most of your ribosomes are not the same as bacterial cells, if you guys remember from last semester, mitochondria have their own ribosomes, and mitochondrial ribosomes are identical to bacterial ribosomes. And so if your cell lets these drugs in and then the mitochondria lets them in, it will destroy the ribosomes inside of your mitochondria. only some of these can actually get into your cells. Most of them are kept out one way or another. But that is how antibiotics tend to help try to kill bacterial cells, or at least prevent them from reproducing so that your immune system can take care of them a little bit better. So coming back to antibiotic resistance, the most common thing that I hear students say that is wrong about antibiotic resistance is, well, I'm resistant to whatever, penicillin or something. You're supposed to be. If you weren't resistant to it, that means that antibiotic would kill you too. And so it's not about you. It's about the bacteria that you have that's causing the infection in you. If you have an infection that's caused by, let's say, E. coli, and the E. coli that's in you is resistant to penicillin and methicillin, that means you cannot use those two drugs to cure you of your infection. Instead, you're going to have to take different drugs that have usually a lower safety margin, so the risk of side effects increases. And then you might have to be on those for a longer period of time. So it's about the bacteria in you, it's not about you. Second half of that question is, what can you do to prevent antibiotic resistance? Number one is, don't go to the doctor every time you get the sniffles. If you have a cold, antibiotics aren't going to help you anyway. So going to your doctor just exposes more people to your cold to get something that wasn't going to help you anyway. There's nothing they can give you for a cold. If you have the flu, they can give you antivirals to deal with the flu virus that is infecting you. Sometimes doctors will still give antibiotics if you are at risk of developing pneumonia from the flu, because pneumonia is often a bacterial infection that attacks people who already had a viral infection. But the antibiotics weren't there to treat the flu, it was there to treat the complication you got from the flu, if that makes any sense. Number two that's up there, if you're prescribed antibiotics, take them as prescribed. If they tell you to take them twice a day, take them twice a day. You also need to finish the entire prescription. For those of you that had me last semester, remember we did an activity with those little bingo chips and the dice, and you saw if you skip even one dose, the chance that you actually kill off all the bacteria in your body is significantly lower. So you need to make sure you take them exactly as prescribed by your doctor. Last one is one that people don't think about, but antibiotics are very often given to pigs and cows and chickens and anything else that we use for meat because it increases their growth rate. So they get bigger and fatter faster when we put them on antibiotics. We're not using them to treat illness. We're doing it to increase the ranchers' or farmers' output of meat so that they can make more money. The problem with that is when we go out into those places and we do testing, we're finding a lot of antibiotic resistant bacteria in farms and ranches, all because they've been using antibiotics not to treat infection, but just to make their animals get bigger. So buy meat that specifically says raised without the use of antibiotics. Organic foods are not supposed to have antibiotics either. It's more expensive to do that, but do you want to have meat that doesn't end up giving you an antibiotic resistant infection, or do you want to pay 50 cents more per pound and then potentially save your life because you didn't get an infection from something that already had resistant bacteria on it? To me, it's worth it, but I understand if you maybe don't have the same income. It's just safer if you can get meat that wasn't raised with the use of antibiotics. How do prokaryotes reproduce? So they can't do mitosis because they don't have a nucleus. That said, the process is actually very similar. They're still going to duplicate their DNA, then they're going to separate it towards two sides of the cells and split the cell in half. It's called binary fission. So that's how they reproduce. That was one of our types of asexual reproduction we mentioned back in the reproductive chapter. What is nitrogen fixation? So in the air around you, the most common gas is not oxygen, it's nitrogen. N2, which that should be a lower, the 2 should be down, not up. But nitrogen gas is not able to be used by plants so that they can make their amino acids and their nucleic acids and things. Bacteria can take the nitrogen gas from the atmosphere and convert it into more usable forms like nitrate and nitrite, and plants can absorb those. And so only bacteria can actually make nitrogen in a usable form that plants can use. We kind of alluded to this when we were talking about crop rotation, when we were in chapter 1 at the very beginning of the last semester, and we were talking about how you could fertilize by doing crop rotation with a pea plant. And that's because peas form relationships with bacteria, and the bacteria that form these nodules on their roots are carrying out nitrogen fixation. Are all bacteria pathogenic? Way big, huge, capital no. Most of them, in fact, are not pathogenic, and a lot of them are actually super helpful for you. That said, usually the only thing people worry about when it comes to bacteria are the ones that are going to cause harm to them. But your microbiota, that's all the species of bacteria that live in or on you that are actually really important for your health. If you try to grow a person in a sterile environment, they end up with really horrible symptoms and autoimmune diseases and increased allergy responses and just all these things that go wrong. So you need these bacteria on you. They also help outcompete pathogens so that you stay healthy for a longer period of time. And so, no, not all bacteria are pathogenic. That said, the next four we're going to talk about are. So, E. coli stands for Escherichia coli. It is a gram-negative rod or bacillus. This is an electron microscope picture of some E. coli. We talked about them a little bit last semester when we were talking about we can make E. coli grow things like insulin for us. I'll come back to that here in a minute too. It can cause food poisoning. There's one strain of E. coli called O157H7 that produces a toxin called the Shiga toxin. And Shiga toxin does really horrible things to your intestines and your kidneys so that you end up pooping and peeing blood because your blood vessels sort of get leakier. This book is about, there was an outbreak back in the 80s. Jack in the Box served some undercooked hamburgers that ended up killing several people, and it was because of E. coli in the hamburger meat. It's why you should never eat ground meat that has not been cooked so that it's well done. It's okay to have a steak that way, although there's still a little bit of risk. But if you're eating ground meat, what happens is as they grind it, the bacteria get all throughout it. And so you have to make sure you cook it at a high enough temperature all the way through to kill all the organism that's in there, or horrible things can happen to you. Also, because E. coli lives in your intestines as well, especially if people don't wipe correctly, they can smear the E. coli from, I'll call it the back door to the front door, and then you get urinary tract infections. And so it's also one of the leading causes of urinary tract infections. Salmonella. So what's wrong with this cutting board is essentially what I want to talk about here. And I think we talked about this a little bit last semester too. But if you are using the same cutting board for your meat as well as your produce that you are going to eat raw, like let's say they were going to use this to make a salad, that means you have just contaminated all of this green and red stuff with bacteria that was on the raw meat that you were about to cook. So now your family eats this stuff, again, let's say as a salad, and then everybody in your family ends up with food poisoning because of the salmonella that was on the raw meat. So never use the same cutting board for meat as you do for things that you're going to eat raw, like salad fixins, essentially. Eggs, same thing. They're almost always contaminated with salmonella, so you are advised against eating raw eggs. And then this one I added in because there are people who decided to have their own like pet snake or pet turtle. They are almost always contaminated with salmonella. So if your kid has a pet turtle and then they play with the turtle and then they just go on about their business, well, pretty soon they're going to stick their finger in their eye or up their nose or in their mouth, and they've just given themselves salmonella. And so make sure if your kids or you have a pet reptile, after you handle the reptile, you wash your hands really well to get the salmonella off your hands. But usually salmonella is a food poisoning issue, and usually it runs its course after a little while, and so antibiotics aren't even needed. You just wait for the diarrhea to go away, and then you move on with your life. Staph aureus, or Staphylococcus aureus is the full name. I mentioned this one earlier. It very commonly causes wound infections. This one is a very common hospital-acquired infection, which means you go into the hospital because you're going to have a surgery or whatever, and while you're in there, the wound gets infected with this bacteria. And the problem with that is if you get it in a hospital, it is more likely to be antibiotic resistant. So if you've ever heard of MRSA, MRSA stands for methicillin-resistant Staphylococcus aureus. And so that means the first two drugs that we usually use to treat staph infections, you can't use them because it's resistant. So now you have to use a more toxic drug called vancomycin, which you also have to have an IV to get vancomycin. So it's not like you can just go home and take some pills. You have to go in and get hooked up to an IV to receive a vancomycin drip. That said, you can still treat it for the most part with that vancomycin. There is also now what's known as VRSA, which is vancomycin-resistant Staph aureus. It's resistant to penicillin, methicillin, and vancomycin. And so you have to use even more toxic drugs to treat that one. And in a lot of cases, you can't treat it. You just kind of have to, unfortunately, hope that it either goes away or do wound care or wait for that person to die from it, because there's not a lot of options for treating VRSA at this point. Bacillus cereus. This is one of our few gram-positive bacilli. Most of the time a bacillus is going to be gram-negative. But you can tell, nice and purple rods, so it's a gram-positive bacillus. The one that this is usually most noted for is, if you ever go to usually some sort of a Chinese food buffet, if the rice has been sitting out for any length of time, this species can be in that rice and replicating in that rice, so that if you now come and eat it, you ingest a large quantity of bacillus, and then you end up with essentially food poisoning. Again, you get diarrhea, you might throw up, you live a couple of days super unhappy, and then hopefully don't eat rice like that again. So don't just let rice sit out on a counter and like pick at it later, because bacillus will very often grow in that. Okay, so now that we've talked about 4 horrible species, remember, not all bacteria are pathogenic. In fact, there's a bunch of foods that I know you guys like that would not exist were it not for bacteria. Personally, my favorite on here is cheese, because I can't go a day without eating cheese of some kind, whether it's mozzarella and Parmesan on a pizza, or it's a nice slice of American on my burger. I gotta have me some cheese all the time. For my people who love pickles and pickle juice has that flavor because it's fermented by bacteria, and so that's part of that too. And so yogurt, most people know it has live and active cultures. That means live and active bacteria inside of that. It's fermented it to break down some of the lactose. And so all of these foods, plus more that I'm not even putting up here, they would not exist were it not for the bacteria that could make that food for us. So remember, not all bacteria is bad. Transgenic, I alluded to these a little bit ago when I was talking about E. coli. So E. coli makes insulin, which is dirt cheap to make, but drug companies have hiked the price up for insulin, making it very difficult for people to afford it. TPA, tissue plasminogen activator, is used to treat or dissolve blood clots for people who have strokes, heart attacks, or pulmonary embolisms. That's made through transgenic bacteria as well. There's a whole bunch we talked about last semester. This is just kind of big two. What's going to happen next for this one? Historically, I've always done these group presentations. Since we are switching to online for this semester, this might be a little bit more difficult, but check in the discussion section. I'm going to assign everybody into a group. You're going to contact everybody through email. I'm going to assign each group its own species of bacteria, and you're going to create a presentation about your bacteria following the format that I've got in the back of these notes. So I tell you what to put on slide 1, slide 2, slide 3, slide 4, slide 5. I figure it'll be easier if I just tell you guys what to make. That way nobody replicates what somebody else is doing. And look for that assignment in the discussion board. Good luck.